maybe I should start over with a little more detail. Why should we save urine before we do it step by step? Why do we have to save urine if it's hardly used? So we have to find a reason why. I claim urine is a better biomarker source than blood. As I said, there's all the other definitions, but I believe this definition probably express or emphasize changes enough. It's a measurable change associated with disease. The change can be protein, can be metabolized, can be microRNA, can be heart rate, can be body temperature, but anything change that associated with the disease is a biomarker. If you play a word game to cross off all the less important one, the most important word left is the change. So biomarker by nature is change. And change is hard to be found in blood because in blood there's homeostasis mechanism, which means in blood all the changes are minimized or removed, so there's less change and less biomarker, especially in the early stage when the homeostatic mechanism is still working. But people may ask, we have so many biomarkers in blood. Why do you say there's not enough? Those biomarkers can only be established when the homeostatic mechanism is out of balance, is out of control. If you have a little high blood glucose at the early stage, you won't see a high level of blood glucose. You have a diabetes is because all your homeostatic mechanism is all used up. You still can't control the blood glucose, and then you see it increase. That's why I believe the markers in the blood is hardly to be early and sensitive one. It may be relatively late stage and when the homeostatic mechanism is running out. So I always try to give an example like we use a lot of vinegars in Chinese cuisine. Like we do dumplings, we use a lot of vinegars. When you drink, let's make it more extreme. You drink vinegars and you drink a lot. What will happen in blood and in urine? Do you see the pH change in urine first or in blood first? And I believe before the blood running out of its capacity, we will see the urine change first rather than the blood. So that's why I believe if this mechanism holds well, we will see more changes more sensitively in urine rather than blood. And in blood, let me give it another example. Let's suppose there is no homeostatic mechanisms. This is against all the physiological textbook. If there's no homeostatic mechanism, you eat something in and they stay in blood. You take a medicine, you take a medication, you take a sleeping pill. What will happen? You will sleep forever. There's no mechanism to remove it. But actually, the homeostatic mechanism is so strong, no matter whatever you take, no matter the body ever seen it or not, they will always find ways to remove it, no matter what medicine. Like proteins can only stay in blood for a few minutes. The half-life is only a few minutes if they are not antibody. Antibody stay longer. But all the other protein drugs are really hard to stay. They are removed very quickly. And another example is like doping in sports. I don't see them really take any sample of blood samples. They can always find a trace amount of changes in urine, no matter what drug you used. But in urine, the good thing is it's accumulate change. But what do they remove? They remove all the useful part for your body. If they're useful, we keep them stay in the blood. We only remove all those that we don't want, and put them in the urine. So in the urine, it accumulates all the changes and at the same time keep the useful proteins in blood remain in blood. I borrowed this slide from the web just to show how the homeostatic mechanism works. There's lung, skin, liver, and kidney. If you drink alcohol, you can remove them from the lung, and a lot of medicines are removed by P450 from liver, and some other things like urea even from skin, and a lot of them from kidney. People can imagine why do we do biomarker study in blood? Because blood connected to all the organs that we wanted to see. We don't want to observe. But urine is not. But actually, urine contains all the unwanted one in the blood. In the blood, it's like a passage, even though it's connected to all the organs. It's the passage. This is the reservoir. This is the place where all the changes stay. That's why I believe maybe we can have like mutations or what kind of for another thousands of years, but I don't see we can have another pouch with such a clean liquid directly connected to blood, which we can use for biomarker research. Here are some of the examples. This is one of, they're not from my laboratory. I hope this can be more objective. And this is from Texas, University of Texas Medical Center. This is one of the examples that with the same people, the biomarker were tested in serum and in urine. This is angiostatin. In blood, they're not performing that well. That's chronic kidney disease, SLE, the lupus, and that's their performance in urine. You see much better p-value compared to the blood. The reason, in the afternoon, I talk about why even the people who work on the blood, they should at least think about urine, the possibility of urine. I did a search on PubMed. When we have biomarker and blood, serum, and plasma, there's 300,000 papers since there's word biomarker, 300,000. Less than 10% with the word urine in it. But actually, the research is done in urine, even smaller, maybe 1,000 paper directly done, not many, which means a lot of people have done this part. They tested if they work in the blood, but they never tested if they work in urine. That give other people a big loophole. Let's imagine you work very hard to find a biomarker in blood. Let's sell the house and file the patent and form a company. What if somebody tests your biomarker in urine, and just like this one, and found that biomarker works even better in urine? All your effort is down the drain. That's a separate intellectual property. But most people stop here. That's why I say there's a lot of opportunities. If you work on serum biomarker, mend the fence. Or if somebody do it for you, that's a big loss. But I encourage everybody who do biomarkers, at least, if you really get addicted to blood, take a look at urine. That's another example. That's 4 biomarkers. They were done in urine, plasma, urine, plasma, urine, plasma, urine, plasma, all at different stages of cancer, I guess. This is done in Seattle and people in Germany. You see all the positive rate numbers. You compare the urine one and the plasma one in each of the condition. This is the only exception. In plasma, it performs better than urine. In all the other categories, This is almost useless, but this one is 100%. There's a lot of, this is done 2009. There's a lot of evidence that for the same biomarker, it is possible that in urine it performs even better than in blood. I don't want to say it happens in all the biomarkers, but at least in some of them. This is actually from Dr. Moses' lab, but Dr. Moses is not here, who worked on the MMPs, and she found that's what she said, demonstrate that urinary MMPs in determining the human cancer both inside and outside of the urinary tract. So even outside of urinary tract. At the beginning, I thought urine is only good for biomarker discovery of urinary system. But actually, there's even more. This is from UK, a biomarker from COPD, lung disease, congestive obstructive pulmonary disease. That's the biomarker performance in urine, U-desmosin. You see the value in urine, the range is 6 to 10. In patients, it's 14 to 22. There's no overlap. 8 to 10, 14 to 22. But if you see the same biomarker in blood, there is an overlap. The highest value in healthy is 0.23. The lowest in patients is 0.21. When there is an overlap, it's really hard to use clinically. So that's a lung disease. I think this is also from Dr. Moses' laboratory, MMP9 and MMP2 in the brain tumor. This is the thing, actually we thought about how far can it go, the condition be reflected in urine. Brain is the furthest place that we can think about. We were trying to design an experiment that happened in the disease happened in brain to see what happened in urine. And one of the postdocs found that Dr. Moses have already done one of the example for us. And that's the one she showed yesterday. Before operation, after operation, this before operation, after operation. The biomarker is gone. There are also other brain ischemia. You see in serum, hardly differentiate. But in urine, there are two different proteins. The difference can be big. And that's the one that I talked about in the previous session. We use anticoagulant to change the function of blood to see where can we detect them more sensitively. In urine, there's more protein changed. In plasma, there's less. Urine, there's more. In blood, there's less. That's exactly the same strategy, same analysis, same machine, and same cutoff, same threshold. Everything's the same. Urine is very informative. When I talk to some of the physicians, that they don't believe there's proteins in urine. They believe if there's protein in urine, people are sick. There's some problem in the kidney. But actually, I have studied that for over 10 years. We haven't seen any person, any healthy person, without urinary proteins in their urine. They all have proteins. And this is unpublished data in the laboratory in my students. And he found that it's already 4,000 proteins if you really analyze very carefully. You really separate them well in the LC, and you can have over 6,000 proteins. And there's thousands of metabolites. I believe there's database in Canada, and there are microRNAs too. And there's actually very low background. And as I compare, I showed, there's a lot of protein in blood, and the change is allowed, not that big. But the urine, we never seen any people died of too many proteins in urine or too many acidics in urine. People will never die, no matter what happened in urine. There's, the tolerance is big, and the background is low, and you can have enormous change. That's the 4,000 protein, the 6,000 protein they analyzed. Interestingly, those are the proteins that enriched. It's not very specific. It's not the protein that only specific to brain. It's enriched in brain. It means most of them, most of the time, most of those proteins stay in brain. But you can see it in urine. Kidney is not even the first one. There's more brain-enriched proteins and more liver-enriched proteins in urine than in kidney. And there's also other major organs. That means if we analyze the urine really well, urine can probably reflect the changes relatively specifically of esophagus, skin, testis, small intestine, duodenum, and those kind of organs. You probably will see changes relatively specifically. That's the one that I try to show. There's, when you do PubMed search, most people working on the at blood and not many people working in urine, and the percentage is very low. That's even just the presence of the word urine. That one actually I talked about. In healthy people, there's a lot of change, which means if urine can reflect physiological changes, it probably can reflect early pathophysiological changes more sensitively. It can probably reflect pharmacological changes very sensitively too, because all those are probably bigger cause than the physiological change. But there's disadvantage of using urine as biomarker. There's all big variations. That's one of the reasons most people stop using it. And so we need a lot of case to validate. We need patients who drink Coke, Diet Pepsi, beer, alcohol. We need all those kind to see those biomarkers are not affected by the beef you eat, by the cheese you eat. So we need a lot of samples. But the problem is urine sample is very diluted and protein can be degraded, even you put them in -80. I can find a paper who showed if you stay longer enough, as long as it's in the aqueous condition, enzyme not completely lose their activity, even in -80. They're still working slowly. So eventually, you will have some kind of degradation. So the way we try to save urine is to remove the most costly liquid, the water. The water is not that informative. As long as we measure the volume, we don't need the water we don't need to keep the water anymore. The water is most costly to save. So the way to remove water is to put it on, go through a filter. This filter is not by pore size, it's by absorption of protein, like nitrocellulose or PVDF. The protein stick to the membrane, and the water part is gone. Here is the membrane. Here is the membrane. When the protein is on the membrane, the way to keep them dormant is by let them dry. When you make it dry, all the enzymes stop working. Nothing can work without water. So the way is to make it dry and seal it in a vacuum bag to keep it dry all the time. As long as you keep it dry, it can stay at room temperature for very, very long. We haven't determined how long it stays yet. We tried for 3 months, no change in the SDS gel. In the mass spec, the proteins are all the same. And think about milk powder. Even without vacuum, they stay. How long do they stay? They stay very long. And for this kind of cheese, you can put it in the drawer or put it on the bookshelf. It doesn't need freezing temperature anymore. Save a lot of money by keeping it. Actually, before, I didn't realize this can be important. I thought about this idea originally for the lab to collect urine samples from very rural area. We can't bring refrigerator there and bring it back. So I said, what about you stick the protein on the membrane and then bring the membrane back? So we dump the water over there so we can work on it. Until one day, I believe this can be even more important than what I said, what I thought about. If we do it even in the hospital, that works even better. So this is a word that I quote from the Mayo Clinic. They are very proud of Henry Plummer, who is the architect of modern hospital. And hard as it to believe today, a single concept developed by Henry at the beginning of the 20th century changed the face of medicine. What's that idea, change the medicine? The idea is centralize the medical record. I guess before that, we all share the same medical record. We probably don't have a separate number. We all have, we probably on the same sheet. It's very hard to see the disease progression. And from this concept, everybody, we have a separate medical record. We can really see how the disease progress. That's the basis of medical research for this century of medicine. And it's now they developed very well. The medical record is paperless, filmless, chartless, but unfortunately, the medical record, which is so important for medicine, is still biological sampleless. We don't have any biological sample in our medical record. We don't have a hair, we don't have a nail, we don't even have a tooth. Even those, they are like non-invasive and harmless, we still don't have. Why? Because they're not informative. I think urine is informative enough and easier to save enough. Just about 1 or 2 months ago, I went to a biobanking meeting, and the organizer asked 3 or 4 of us at the podium to say what's the future of biobanking. I thought about, I think ideally we should preserve easily acquired, non-invasive, informative samples long-term at room temperature. This is about ideal. I think that URIMAN serves the purpose. It can be easily acquired, non-invasive, very informative, and can stay long-term at room temperature without any special storage condition, and it's cheap. So overall, I think urine smells good. And if you try it in the lab, work on it. Don't drink it. And that's the people who work on it, and the funding from Ministry of Science and Technology of China. And I hope I can save more time for questions. And the graduate student in the lab, Fan Shuang, will show us a video that they made how exactly that urine is urinary proteins and nucleic acids are saved on the membrane. And I don't know, should we have question and answers first, or later to show the? Oh, sure.
When you get the membrane, you let me wash that, because a lot of salt may be in the membrane as well. Then you get the membrane to the water, and the salt will come back and create more protein.
Most of the salt should stay with the liquid. I believe, yes.
And you don't wash it.
Yeah, we don't do it. Yeah. Okay, maybe Fan Shuang will come to show us the video.